Oh my God.
Okay. Sorry. Polysaccharides are like glycogen is a polymer of glucose. It can be broken down into the monomers via hydrolysis, and then you have lots of glucose. Where is glycogen stored? Somewhere in your muscle cells. Good. Okay. There's other polysaccharides that could be broken down. Starch is an example of another one. All right, other 6-carbon sugars could be modified using existing enzymes and turn them into glucose. Both of these would start at the beginning of the Krebs cycle. Glycolysis words. So galactose, fructose are other 6-carbon sugars. Okay, so proteins could be broken down to their amino acid components, and then you'd have to do some other stuff. So here's a carbon chain we'd want to utilize. The amine group would have to be cleaved off and disposed of, and what do we turn our nitrogenous waste into? Like us? Urea, right. But depending on the organism, it'll convert it to ammonia, uric acid, urea, depending, and that will be just excreted. But the carbon skeleton could be modified in existing ways and could either enter into glycolysis or the Krebs cycle, depending on what it's converted into. Okay, fat. Remember we talked about fat store twice as much energy as carbohydrates? And you're going to see why, like right now. What happens is they're cut off into their glycerol and fatty acid components. And then the glycerol, which is the gray box over here, is converted to G3P. And that enters into, after that 5th step of glycolysis, right? Okay. And what's interesting is that yields more energy by itself than glucose actually does, because you don't have to initially invest the ATP to make the glucose unstable to split. Does that make sense? Okay. And then look what happens to the fatty acids. They turn into acetyl-CoA groups. So they're cut into 2-carbon groups. Look at all those. Look at all those 2-carbon groups you can get out of this particular fat. Each one of those will enter into the Krebs cycle as acetyl-CoA. It does require a little bit, but the initial input would be nothing compared to, like, for this example, the amount of electron carriers produced to make ATP. So it might, but enzymes could also catalyze it and not necessarily need the ATP. So that's a lot of energy that can be released from just one fat molecule. Look at all those acetyl-CoAs you can make. A ton. And fats can have different length carbon chains in their fatty acids. So there we go. All right. This is a picture of this in your book, just kind of showing the different places that, like protein, for example, could enter the fat, and then the different carbohydrates. So it's important that all of this is coordinated. What happens if, like, the Krebs cycle is running faster than the glycolysis can go? All right, so you want everything kind of going at the same rate, and there are ways to sort of slow things down or speed things up a little bit to make the rate sync between all the different stages. Don't really need to know. I want to talk about. So digestion is breaking down everything. We don't need to know this. Well, I already talked about it. Okay. What's interesting is that this can kind of go backwards actually to help synthesize different molecules, because enzymes can always catalyze the forwards and backwards reactions. Obviously, if you're going to go backwards, it would require a lot of energy, but it can happen. It can happen, and it does. Sorry. I used to have these giant posters in the old Spring Valley, like eons ago, that had all the different reactions in the different macromolecule or biomolecule pathways. This is what we call carbohydrate metabolism. These are all the different reactions. This one here, right here, is what we talked about, glycolysis and the Krebs cycle. But look, there's all kinds of other things that will help to build or break down different carbohydrates. Gluconeogenesis is just the catabolism of glucose. Lipid line, notice here is the Krebs and glycolysis. Here's everything else. Complicated. Amino acids, polyG. That was a huge poster. Anyway. 8, you have to get in your diet. They're already synthesized because we can't make them. We only have the enzymes to help make 12. And these are the ways you help to synthesize the different nucleotides. All this other stuff over here. Okay. Acetyl-CoA, why do we care about it? Acetyl-CoA is a really important regulatory molecule. And let's talk about it. Okay. Well, we'll talk about it. All right. Remember feedback inhibition a long time ago? Yes? Yeah. Okay. So when a product of the pathway can go back and allosterically inhibit an enzyme that occurs earlier in the pathway. You remember that word, allosteric inhibition? Or regulation is when it binds to a site other than the active site and change the enzyme shape to make it either work better or not work at all. So inhibition would be preventing it from working, obviously. And obviously it makes sense if you have a whole lot of like G, for example, in this picture, you're going to have a lot of it sort of moving around inside of a cell, and it's much more likely to bump into that enzyme earlier in the pathway. And if it does, it can bind to it and turn it off until the G's been used up. And then this binding would be non-permanent, and so it could just flop off and then the pathway could go again. And it's a way for cell to save energy or save resources because it prevents, it's like your wheels spinning when you don't really need to go anywhere. You don't want to waste gas. All right, an important enzyme, the only one I require you to actually know in the whole thing is something called phosphofructokinase. Phosphofructokinase. The kinase enzyme is something that phosphorylates things, remember? The chapter you're going to read about when I'm not here on Tuesday, it'll probably hurt your head, is chapter 11. It won't be anything like you've ever heard before. Sorry.
Are you going to come back and explain
it? I'll teach it to you, I'm going to say, yeah. No, I'm just going to make no.
How important?
Good luck with that. No. Anyways, we'll learn about more kinases on there. The phosphofructokinase, it helps phosphorylate phosphate into this phosphate. So anyway, but why do we care about it is because it's the main enzyme that's allosterically regulated to help keep the glycolysis and the Krebs cycle going at the same rate. Yeah.
Have to be defined.
It usually is, and not necessarily. And you'll see here it's not necessarily. Why this particular enzyme? It's because this is the one that phosphorylates the glucose intermediate the second time, and that makes it super unstable, and then it splits into 2 pieces. So if you stop it from phosphorylating it the second time, then it's not so unstable that it won't split. Because once you split it, it'll just move down the second set of steps.
Is it after it becomes like glucose or something?
After it splits?
No, like that enzyme after it becomes fructose, the glucose.
It already is fructose 6-phosphate. It becomes fructose. It became the fructose in one of the first steps of glycolysis, where it's sort of like sunrise. Now, what actually increases the activity of phosphofructokinase, what makes it go faster is AMP and ADP. Why does that make sense?
There's a larger amount of ADP that means the ATP is used at a faster rate.
Yes. Did everybody hear that?
No.
Say it louder.
If there's a larger amount of ADP, that means that the cell's ATP is being used at a faster rate.
So if there's a lot of ADP in the cell, that means the cell has used already a lot of its ATP. It means it's low on ATP. You need to make this whole thing go faster so we can make more ATP. Now, AMP is adenosine monophosphate. It's like even some of the ADP's been used. So AMP only has one phosphate group, ADP has two, ATP has three. Make sense? So both of those will make this whole thing go faster because that means the cell's running low on ATP. Okay. Now, what can actually inhibit phosphofructokinase is ATP. And now, in this case, Jackson, it is sort of the end product of the whole thing. And if you've got a lot of it going on, you can slow it all down a little bit. What's also interesting is that citrate will inhibit it. Citrate is that first product of the first enzymatic step of the Krebs cycle, where the acetyl-CoA binds the oxaloacetate and makes citrate. Citrate is that 6-carbon compound. You remember? Okay. So if there's a lot of that, maybe it's going too fast, and you need to allow the citrate to kind of move through the Krebs cycle and, be broken down. And so it inhibits phosphofructokinase to slow glycolysis down just a little bit. So it's just important to keep it all running at a steady rate so the cell doesn't waste energy. Questions? The only enzyme I want you to know, and I want you to know how it works in terms of regulation. I already talked about all that. Okay. The end. Okay. Now we talk about photosynthesis. Oh wait, no, we're not. I forgot. I want to talk about this. Your extra credit. Most of you got it right ish. So I'll talk about it. So yeah. Oh yeah. Yeah. I always get partial credit. Like I learned it from my mom. My mom was a physics teacher. She would always get partial credit. Like if you got the answer wrong, but then you did all the work the right way, she'd like only take off the line. Nope. But she made it so that people could do work. Anyway. Okay. So people raise cows for a lot of things, like Andre. They have cows, right? Because they sell them for Angus beef. He is fragile, anyways. But also they kill their cows, but he doesn't kill them. Someone does. But some people have cows and they make milk. Those cows don't die. They do eventually. Anyways, so why do we make hay? Because cows eat the hay. It's nutrition for them. Help them live. Look at those little cows. Yay. Okay. Lots and lots. So cows eat something called alfalfa hay, but they sleep on a different type of stuff. A lot of times, like at the zoo, you see there's like the stuff on the ground that they're sleeping on. You're like, that's hay too. It's not exactly hay, so it's straw. They don't really eat that. But anyways, alfalfa is grown. It looks like this, kind of pretty. So there's fields of hay. You might not know that what you're looking at is hay. There's a place on my way home if I go the back way. Kelly Mill Road, maybe is that what the road is? There's a big. The land used to be for sale. It was like after you pass over Hardscrabble, you're going like towards the house. Yeah, there's this big, like the basketball player's house is on the left, big, and then the one on the right, like there's big fields. Like at some point you'll see like hail days, bales of hay, because they anyways. But that's basically alfalfa day. Sorry, I can't talk. My brain goes faster than my mouth does sometimes. Anyway, so then you cut the hay using the big tractors. And then you have to dry it out and just sort of sit there. And then you can bale it. There's actually a big giant machine thingy, like a tractor thing, attachment that will bale the hay for you. You have to make sure the hay is dry. And there's actually a certain percentage, like it has to be more than 15% or less than 15% like wet still. And there's a reason why I'll talk about. So then they go around and like bale the hay, and here's a baler. That's what it's called. And then they leave the bales. And they stack them. And they store them in silos or barns. And they can sell it for a lot of money to people that grow cows. But what happens if it's not completely dry or mostly dry? So hay is made up of plant material, which has a lot of sugars in it. And sugar and water, okay, is actually a really good source of food for bacteria. Okay? And particular bacteria called mesophilic, which are heat-loving bacteria, can grow in this environment, and they'll undergo respiration. And they'll actually, a byproduct of respiration is, remember, heat. And sometimes even the hay itself, some of the cells in the hay can still undergo respiration as well and also produce some heat. And actually, you'll start to see the bales of hay start to release like steam. And then they actually could spontaneously combust, really. And people can lose lots of money, the people that do this. And it can destroy your property and all that kind of stuff. So that's why you don't want to have wet hay. There you go. And then the milking, look, I would not want to. I used to want to be a veterinarian a long time ago, and large animal vets. Does anybody want to be a vet? No, okay. Large animal vets, people would be like, what do you want? I was like, small animal, like cats and dogs. That's all I ever want to do. Like, no exotics, no like birds and lizards and stuff. I'm not that kind of person. And no large animals, because when you go to vet school and have to do large animals, you have to like stick your entire arm up their butt. Like, literally.
Yeah.
Huh?
Give us love.
No.
I know, I just, no.
You do. Yeah, but
What's the animal that you got to stick your arm up there?
Horses and cows.
Oh.
Wait, more than
If you're a large animal vet, you do horses and cows and donkeys and, you know, stuff. Anyways, okay. No, thank you. No, thank you. Okay. Okay, so I'm going to start photosynthesis, and to be honest, I don't really want to give you a note for today. So I'm going to give you one tomorrow. Okay, so hold on to them. If you didn't finish, or whatever. So you should thank Tori for this, because I don't want to stop the video. It's really what I'm doing.
Thank you, Tori.
But, okay. Plants, mostly, well, this chapter is about photosynthesis. Hopefully you read it, and you maybe remembered some of it from Bio 1, right? But plants undergo photosynthesis and respiration. So photosynthesis is how they make their food, okay, how they make their sugars that then they can use for cell respiration, right? They're connected, see? So anytime you see a plant that is green, like in this picture, the green parts of the plant are the ones that are photosynthetic. Now, plants are the only things that are photosynthetic. Some bacteria can be photosynthetic as well, okay? And there's things called chemosynthetic organisms that can make sugars using chemicals, not light energy. So anyways, but we're going to focus on photosynthesis. I'm going to show you some other stuff, pictures, because I was reminiscing earlier.
Beer bottles out of plastic.
What?
Wait, if we are going to we're going to start drinking your content.
But you can taste like
It was so good. It worked so well. But it was really good. Yeah, I thought we
I just had pizza last night.
I was happy.
There'll be no more.
Okay. A long time ago, I had green parts of plants, but synthetic. These are just pretty pictures of plants after sharing. Has anyone ever been to London? Yes, there you go. There's a place in London called Kew Gardens. That's pretty famous anyway. These pictures are all taken in Kew Gardens. I took a lot of them at the time for AP biology way back when, because we used to have to learn all about flowering plants and non-flower plants and plant reproduction, which you don't really need to know anymore for AP biology, but I still think these pictures are cool. So, look, pitcher plants. We will talk about pitcher plants. You know what they are? No. Pitcher plants. Does anybody know?
They hold water.
They're like. Okay, so what happens is that, sure, but little bugs, flies, will crawl up here, and their walls are really slippery, and the fly will like go in and try to get some of the nectar, like, and they'll fall to the bottom, and there's hydrolytic enzymes down there, and they actually get digested, and the pitcher plant absorbs the nutrients from the flies. They're kind of like parasitic plants. They're not animals, they're trash. Kind of, yeah. These are pitcher plants. They're neat. They're really big leaves. This whole thing is like photosynthesis. Large surface area to get the most solar energy. Yeah, that's huge. I know, plants are really cool. I love plants. These are pretty flowers. Yeah, oh, there's Kew Gardens. So I tried to find the pictures from my honeymoon, but I couldn't find it. Yeah.
Do our plants have seeds in it? Looks like it has.
No.
No, okay. We were doing Jeopardy and there was a question about, yeah, that's what I thought. And then we said the answer, it was something about like how do they, like one of their, I don't know what it was, but the answer could have been spores or seeds.
Yeah.
But they told us spores was wrong because it didn't include all plants. But then we were like, seeds don't include all plants.
Well, yeah. So bryophytes don't have seeds, and there's other plants. We'll talk about that when we talk about plant evolution.
I was just mad because their answer was wrong. They were like on Saturday at Columbia.
Yeah. The advent of the seed was a thing that happened along plant evolution, but not all plants are seed-producing plants. Yeah. Okay, that's all my pretty pictures from. Okay. When you Google words, you get weird things. Something I got. The water is like, I don't know. TheAwkwardYeti.com. Okay. So, there really should be food from lightning here. But we're going to be specific. Okay. So we know all this. Yes? Consumers, producers, heterotrophs, autotrophs, explorers. I hope you know it all. Okay. But I'm going to find my next one.
Oh my God.
Did the shot drawing super really help you?
Yeah.
Like a lot.
Yeah. Do what?
Is it like a separate lab or is it drawing it?
Separate lab. What do you mean?
It's like, is it like a lab, like last time?
There's a lot of questions you have to answer. Okay. So photosynthesis, organisms that photosynthesize are the producers of an ecosystem. They produce the sugars that other organisms like eat when they eat producers. But anyway, so these would be autotrophs. We're talking about plants. So they are connected. We consumers eat those producers for those same organic molecules that producers are producing. So, that was redundant. So remember, we just talked about cell respiration. So it's the slow oxidation of glucose to eventually produce carbon dioxide, which is released in the atmosphere. The cells make ATP, heat energy. There we go. Uses oxygen. So if you actually, this very exergonic process, right? Photosynthesis is the exact, almost opposite reaction. It goes the other way. So it utilizes carbon dioxide, utilizes water, utilizes solar energy, there's energy in the photons to produce glucose and oxygen gas. Some heat is also produced as well. And it's a highly endergonic process. Requires a lot of energy. So we say the free energy of cell respiration is negative 686 kilocalories per mole. Remember when free energy is negative, it's exergonic, okay? So the free energy change for photosynthesis is positive 686. It's like the exact opposite.
Not so relevant. How do plants that are in the ocean get fresh water, or like how do they get rid of the salt? Or
So they're isotonic to the seawater, so they don't have to worry about it. Or they would. I don't think being turgid is like a big deal in plants, like saltwater plants. But you need to know that not most plants are photosynthetic, not all. Some plants are complete parasitic plants, and there's a picture, I don't remember what it's called, but it's a plant that looks really white. It's like the whole thing's white. It has no chlorophyll, and basically what it does is it puts little basically kind of like roots or whatever into a host plant and steals the sugars out of the host plant. Mistletoe is a parasitic plant. Mistletoe kind of does the same thing. You ever see it like in the wintertime? You look up at trees, you'll see mistletoe in trees. It's a parasitic plant. It's stealing phloem sap. Okay. So if you are a plant, then you have to wait to collect light energy. You have to have a way to transform it into chemical energy of some ATP and NADPH that eventually makes your glucose. You have to have a way to store the light energy so you can move it around, and that would be the glucose molecule. And you need to get the building block atoms to make the glucose from your environment. So this would be carbon, comes from CO2, the hydrogen, the oxygen, nitrogen, potassium, sulfur, magnesium, all the stuff you need to make whatever other organic compounds the cell needs to make. A lot of these come from soil. So the soil is highly important as well for the health of the plant. So the water comes from the soil, the magnesium, sulfur, potassium, phosphorus, nitrogen, all that stuff comes from the soil. You ever notice like fertilizer, has NPK ratios? You may know what I'm talking about. You're just staring at me, so if you don't know, okay. Anyway. And plants produce all the organic molecules they need, all of them. They produce G3P actually only from photosynthesis, so they can combine two of them together to make the glucose, and then they can do whatever other stuff they need to make all their nucleotides, all their amino acids, all their everything. Plants are pretty cool. Okay, so let's look at a plant real fast. I think a long time ago I said plants are kind of different because they have two different systems. They have an above ground and also an underground system. The above ground system grows against gravity. You're going to see this is the part where you're going to have the green parts of the plant. Now this is a tomato plant. Even the stem on a tomato plant is green. So all the green parts, like I said before, undergo photosynthesis. But something like a tree, the giant tree trunk is brown, right? That does not photosynthesize anymore. Only the young, like the leaves that are green, even like some of the branches and stuff are all woody now. So the woody parts of plants don't photosynthesize, just the green parts. The underground system, notice, do you see any green under there? Why not?
No.
There's no photons, right? So it can't photosynthesize. So under there, these roots down here, they undergo cell respiration. So they can continue to make ATP and live. But they can't photosynthesize. So the shoot system, which is the above ground part, sends the sugars to the root system. So the root system can then use it to undergo aerobic respiration. Okay, so that's why if you cut off all the green parts of a plant, on a tree, like you cut off the tree trunk, it won't grow back. You know what I mean? You don't have to like take the roots out necessarily, kill it, because then there's no more green parts in the sun.
What about when people are going on there, they like cut off all the branches of the trees? So it's like studies?
Okay, so that's different because they leave meristematic tissue. I'll tell you what that really means. So plants are also cool because they have indeterminate growth, which means that we mature and then we don't have any more stem cells except for like our blood ones. Like if I cut off your arm, you can't grow back. Okay, but plants, if you cut off their arms, as long as there's some meristematic tissue, they'll grow back. The meristematic tissue is tissue that is able to differentiate into anything. Anything. And a lot of times there's meristematic tissue, like as long as you cut off a branch.
Looks like stem cells.
How you spell meristematic? As long as you cut off the branch like right there, and you leave this little node, like right here, this little thing right there, there's meristematic tissue right there. And when it becomes spring then it'll grow back. The reason they do that is cut off what's called the axillary. Okay, I'll just we used to learn all this when we did our big plant unit, but there's something called the axillary bud, which is the very top part. And plants, their goal is to get more and more and more sunlight. So their nature is to go straight up towards more sun. Okay, so there's something called the axillary bud at the very top. That as long as it's there, present, then the whole thing will keep growing up and not out. So it'll grow a little bit up and mainly up. If you cut off the axillary part, the top, then it causes these other nodes that have meristematic tissue to become activated, and they'll start growing more that way. So the plant will then grow out and then up again that way. Have you ever noticed some people are pruning their plant, they like cut at the top moss and allow them to get more bushy? That's why. Anyway. So that's why you can prune them and they'll come back, because that meristematic tissue is there. But if you cut off the whole thing, like it says, like down here, there's no more meristematic tissue down here. What's this stuff? I don't think I was done with that, I guess. Okay. So the sunlight is collected mainly by the leaves. The leaves have a big surface area, and they're the major solar collectors. But any part of the screen can photosynthesize. The CO2 comes in through tiny openings in the leaves called stomata. I remember learning that in Bio 1? Yes. Stomata. Stomates is plural. But they're little tiny, I have like slides on stomata a little bit later, but this is where the carbon dioxide comes in and oxygen escapes for leaves. Water comes in through the roots, as well as all those other nutrients like nitrogen, magnesium, iron, all that. Comes in through what's called the soil solution around the roots. This picture reminds me always of the Little Shop of Horrors. You know what I'm talking about? What? Little Shop of Horrors? Yes. You know what I'm talking about?
Little Shop of Horrors.
Oh.
Baby shop.
Okay, I didn't say horse. What? That's what I said. Does anybody know what I'm talking about?
Yeah, I know.
You know what I'm talking about? It's a musical, and it's about this plant. That plant, he like starts talking and he starts to eat people. It's really funny. They made a movie in like with Greg Moranis. Honey, I Shrunk the Kids. He was the yeah, that guy. He was in like they made a movie. Life in High School did that play like a couple of years ago. Anyways, like it's really funny.
To be human? Does it have to be mine? When am I supposed to get it? Missy Mo. Get all back now. Missy Mo. Feed me all night long. How big and Missy Mo. I can't grow as strong. You eat blood on me too, let's face it.
Okay, it's really funny. No. Anyway, but that picture of that tomato reminds me of the plant. Anyway. All right, so Mata, like I said, opening. It's the actual opening itself, though. It's not the cells around it. So do you see, like in this picture, the little red parts? Those are actually two cells. They're called guard cells. And the guard cells are what regulate the opening or closing of the stomata. The guard cells, when they become very turgid and full of water, they actually fill up, and because I'll talk about this in another chapter, but they have cellulose in their cell wall wrapped around in a certain way that when they become turgid, they bow out and look like lima beans and allow the opening to happen. And when they become flaccid, they lose water, they close like this. Anyways, so when there's lots of water, the plant is not, in danger of losing too much water. The stomata can remain open and they can exchange gases. So that's where that happens. Some other parts of a plant you need to know. Oh no, this is fine. Label. Interactive lecture today.
Remember those colorings?
Oh, and look. Oh, it's compounded. What happened to what? Skeleton got started.
Oh no!
I had them all rolled up in the back.
Miss Mike was at Columbia College lab, had like all of the like things you had, they have like all the same things.
Oh yeah?
She had like that little human and like the posters on.
Oh, cool. There's the front and the back. So right now we're doing, you can label some of the parts on the front. The front. Did I have one? Did you get one? Okay, so here's the leaf. Figure it out. So let's talk about it. The cuticle is, you ever notice leaves look kind of waxy? Yeah. Okay, that's there to help protect the plant. It's called the cuticle. All right, you can see it labeled here. And it's the very top part, and right down here. Don't get ahead of me. Do cuticle and listen to me, because I'm going to say stuff, and you need to know it. The cuticle is highly hydrophobic. It's wax. And it helps to, one, not only prevent desiccation or dehydration from the plant, but also it's there to protect the barriers that prevent insects and things like that from eating it. They can obviously still eat it, but it's there as an initial protection, also against bacteria and stuff. So that's the cuticle. Notice it's very thin. That's why plants look waxy, kind of. All right, and then right underneath the cuticle, you're going to see a first layer of cells. This is called the upper epidermal cells. These are here. Here. And there's a lower, so top and bottom of the leaf. I guess we don't talk about it. And then let's move down from the upper epidermis, and we move into these tall cells. They're called palisade parenchyma. Everybody say parenchyma. There you go. Most of the time, I usually forget to do that, and I tell you to say it yourself. I always say parenchyma, it's really awkward. Parenchyma. Anyway. But this is the palisade layer of mesophyll cells. Is really there's mesophyll. You can just say palisade. Anyways, they're long, like columns of cells. And then you move down into what's called the spongy mesophyll. Why do you think this is called the spongy mesophyll? It looks like a sponge, like with lots of openings, right? You see all the openings? There's a lot more space in here, kind of like a sponge has a lot of space in it. Okay. Also within the spongy mesophyll area, you're going to see the vascular tissue of the plant. The vascular tissue of the plant is sort of analogous to our vascular tissue. It's where the sugars and the gases and the water and all that stuff, well, not the gases, I guess the water will travel. So here's the vascular tissue. The vascular tissue of plant is xylem and phloem. So that combined is the vascular tissue of the plant. And I learned also this in 9th grade from my teacher, Mrs. Johnson. Xylem up and phloem down. So what comes up from the root? No, well, yeah. But what's inside the xylem that comes up from the root? Water. And the like nitrogen and all that stuff. So what the xylem carries is the water and the soil solution stuff. Okay, xylem up. And phloem down. So what do you think is produced in the leaves that will travel other parts of the plant? The sugars. So the phloem, we call phloem sap, is highly sugary. It's actually where you get like maple syrup from. You put on pancakes, they drill holes in the maple trees and put these little like buckets and allow the phloem sap to just fall on out in the bucket. And that's what you eat. So. Well, they don't kill the trees, because they allow some of it to travel down, but they take some of it. So that is the structure of a normal, what we call C3 plant. I'll tell you what that means. Well, you read about it, right?
Yes.
Normal C3 plants have this structure. We'll talk about the other structures later. So there you go. And then you'll notice the stomata be located down there. Opening. Good. Did you like what?
I think they're all northern.
I don't know. Yeah.
Okay.
Where we haven't gotten to. Okay, you can stop at like nucleus, chloroplast. I haven't stopped like any of that yet. I had to leave cuticle, epidermis, and mesophyll cells. That's all I had to do right now. And actually, you should make sure you know the palisade versus spongy.
What?
Palisade versus spongy mesophyll. Okay. Whoa, sorry. Remember transpiration? What is that? How water moves up a plant? Yep, up and out. So let's talk now about the structure of chloroplasts. Chloroplasts are the organelle of plant cells that actually undergo photosynthesis. So if we look at it, chloroplasts have a double membrane on the outside. Both membranes are smooth. Okay. Inside the inner membrane is a fluid in this space called stroma.
Do you know what that number is made of?
Top of the bilayer. I'll tell you earlier. The outer membrane. You'll need to know the outer membrane. We'll talk about that in a little bit. But the inner membrane is there. Okay, so let me show you. So outer membrane, inner membrane, and then there's fluid surrounding the green stuff. The fluid surrounding the green stuff is called stroma. The stroma is where the enzymes are located for the Calvin cycle. And notice that, what's the green stuff in there? That's called the thylakoid membrane. Okay, that's actually a third membrane of chloroplast. It's highly convoluted. It's formed into these little discs called thylakoids, and they're stacked up like a stack of pancakes, and the stacks are called grana. Okay, they're actually, I mean, they look like pancakes, but they're more like pita, because they're hollow inside. Each thylakoid is like a pita. It's hollow inside. There's fluid inside called the thylakoid space. Is this all ringing a bell from Bio 1? Okay, good. All the thylakoid membranes are interconnected. You can kind of see the little interconnections in here in this picture. But the reason why it looks like it does is to have a lot of surface area. It's sort of analogous to the highly foldings of the mitochondrial membrane. It's just differently, done differently here. The thylakoids and the thylakoid space all are where the light-dependent reactions occur. The Calvin cycle is what we call the light-independent reaction. Why do plants look green? Because of the chloroplast. So why do chloroplasts make the plants look green? Because of chlorophyll. Chlorophyll is the main pigment that absorbs all the other wavelengths of light except for green, right? We'll talk about that later. But oh, there's stroma. The stroma is also where the chloroplast DNA is located and ribosomes are located. Remember how we talked about that a long time ago that mitochondria and chloroplast have some of their DNA? It's in the stroma. Okay, the thylakoid membrane also contains not only chlorophyll, but electron transport chain and an ATP synthase enzyme. All of that's in the thylakoid membrane itself. So that's a thylakoid up there, and the yellow is a thylakoid space inside. Oh wait, just kidding. There's our thylakoid. Hydrogen ions go actually inside the thylakoid. They're pumped into it. It's kind of opposite from a mitochondria because the hydrogens were pumped out of the matrix. Okay. But because there's an ATP synthase, that hydrogen ion gradient is generated to eventually make ATP. That the ATP made in chloroplast is used by the light-independent reactions, by the Calvin cycle, to make the glucose. It's not used by any other parts of the cell. The reason a lot of times people think photosynthesis is a little bit easier than cell respiration is because we've already talked about how a lot of this stuff works already. Like you know how ATP synthase works. You know how an electron transport chain works. So we just kind of have to put it together in a different way. On the back of your sheet that I just gave you is this diagram. It's just kind of sideways. Okay. It's kind of sideways, like here. You see it? Diagram B. Okay. You can go ahead and label it right now when I talk about it. Okay. So here we have, I like this diagram a lot in your book. It's kind of analogous to the one that they use for cell respiration to talk about all the different parts and how they're connected. This is a good one for photosynthesis. So over here we have light energy and water are the two things that are used, that come from the environment, that are used by the thylakoid to undergo the light reaction. Okay. The product of the light reactions is oxygen gas, the product that actually leaves. So oxygen is the byproduct of photosynthesis that's not used, so it comes out. Now the two main products of the light reactions that are actually important are these two right here, ATP and NADPH, sorry. And those, notice, are used directly by the Calvin cycle. So the thing over here is the Calvin cycle that happens in the stroma. It takes in carbon dioxide from the environment. It uses the ATP and NADPH made by the light reaction to produce a sugar. Now, the direct product of the Calvin cycle is a G3P molecule. You need to know that. Glucose is not the direct product. It's G3P. Notice, I guess I didn't mention these up here, but the light reactions also need NADP plus and ADP plus a phosphate, because then they make NADPH and ATP. What is NADPH? Or what is NADP plus? They're analogous to NAD and FAD, right? They're electron carriers. They carry electrons. And in this case, they carry high energy electrons, because the point of the Calvin cycle is to slowly reduce carbon dioxide, give it high energy electrons, give it hydrogen. The electrons come and hydrogens come from NADPH. So yeah, the other things in here, so the top right would be a stack of thylakoids called granum, and then it's really just pointing to the Calvin cycle. Oh, I guess these are just the labels written up to the side. So you didn't have to write it in the thing. That's why. Okay. The light reactions, like I said, occur in the thylakoids. They're also called light-dependent reactions because they need light in order for them to run. Without it, they do not run. These are the reactions that trap the energy and convert light energy into chemical energy of ATP and NADPH. And again, NADPH is like that electron carrier, keeping your money in the bank. So sort of for the Calvin cycle. Calvin cycle is also known as the light independent reactions. When I took Bio 1 forever ago, I learned them as the dark reactions, so that's not the best way to say it. But even if you Google it online, you still might find this. The dark reactions, we don't like to say that because they won't occur in the dark. Okay, they just don't need light in order to happen, but they won't occur without the light reactions. So actually, they need light, but just not directly. In order for the Calvin cycle to happen, you have to have ATP and NADPH. In order to make ATP and NADPH, you have to have the light reaction. Does that make sense? So it doesn't need light directly, but it definitely needs it indirectly. So we don't say that anymore. These are the reactions that actually produce the sugars. Slow, very exergonic, produces slow reduction of the CO2. Yes, what I was saying. Sorry, yes, it is highly endergonic, sorry. Yes. Got it? I can definitely tell from my neck. Oh, the rain. What? This? Wait. This. It says CH2O. It's a sugar. I don't know why it says that. All right. This picture is showing a mitochondria as opposed to like compare with the chloroplast. Notice the brown areas, like the dark brown, is where the hydrogen ion gradient is built up. So it's highly acidic, right? Notice in the mitochondria, the hydrogen ions were pumped out of the matrix into that intermembrane space. In the chloroplast, it's the opposite. It's pumped out of the stroma into the thylakoid. You see that? Okay. They both have electron transport chains. They both have ATP synthase. So they're very similar in terms of how it creates a gradient and what it uses it for. but it's different. They both have ETCs. Another difference is an electron carrier that's produced in the ETCs in the chloroplast is NADPH and not NADH or FADH2.
What translates from like the lost energy from the electrons from the NAD?
Say that again?
Like the H ion to create that gradient, it's still
Yeah.
transferred the same way?
Yes. We're going to talk about it. The electrons get highly excited with energy from light. And so there's two parts. There's two photosystems in the light reactions. Have you read about it? Photosystem 2 and 1. The light energy absorbed from photosystem 2 is used to generate the hydrogen ion gradient. Photosystem 1 excites the electrons that are going to go into NADPH. We'll talk about it. Okay. Here's the ETC you're familiar with from cell respiration. And remember, the electrons are already highly excited because they came from glucose, right? They're already really excited. And they're just dropped off here. They slowly lose their energy to create that hydrogen ion gradient and then slow back down to make our ATP. Yes? Already highly excited because they came from glucose. A little bit different. The electron transport chain of photosynthesis is a little bit different because the electrons here are not already highly excited. The light energy is what makes them highly excited. And tomorrow I'm going to kind of show you. I got to go get some spinach, but I'm going to show you what happens when you excite electrons from spinach. They fluoresce. Anyways, can't do it at home without. Well, I'll show you. Anyways, the light energy makes them highly excited. Then they flow down a little tiny ETC. This is the ETC I'm talking about. Hydrogen ions are pumped in, and then they get highly excited again over here, and then they're put into to make NADPH, which carries those high energy electrons to help produce carbon dioxide. We're going to talk about this in way more detail than you probably ever want to know. This is just an overview. So ATP synthase does the same kind of thing here. That's all I want to say. All right, so let's talk about chlorophyll. There's a number of different pigments that are in plants. Right now you can see actually a lot of the pigments you hardly ever see, the carotenoids and xanthophylls, because some leaves are turning yellow and orange right now and even red because the chlorophyll is breaking down right now. And the chlorophyll is going away and it's actually exposing the other pigments, which is why leaves are appearing red and yellow and orange right now. But the main pigment is this one, chlorophyll. Chlorophyll has this structure. What's interesting is that up here is highly polar, down here is a fatty acid tail. So this molecule is what? That's firm. Amphipathic. It's both polar and nonpolar. It makes sense because it's located in the thylakoid membrane. So it has a polar part that's where the phospholipid heads are, and has a fatty acid tail where those phospholipid tails are, right? It's in the membrane, so it makes sense. Notice in the very middle of chlorophyll is magnesium. Without magnesium, plants can't properly synthesize their chlorophyll, and they don't grow very well. They kind of are pale-ish, not so green, so they need that. There's two types of chlorophylls, A and B. They're both very similar in all this except for up here. There's one little area, and chlorophyll A it's CH3 and chlorophyll B it's CH0. You don't really need to know it. I'm just showing you the difference. Up here, this ring structure is called a porphyrin ring. It's the area that the energy from the photon is actually absorbed. Electrons are excited in. The pigments, or particularly the chlorophyll molecules, are arranged together in something called a photosystem, which I'll talk about in a second. Hold on. Let me see if I have the others. I'll get to it. Okay, good. Okay, we'll talk about it. Okay, so I remember this from, I don't know, when did you learn this? Physics, physical science? The electromagnetic spectrum, yes? Okay. The part that we care about for photosynthesis area in here called the visible light spectrum. Okay, you go to shorter wavelengths way down here. Okay, so remember, I'm just talking about structure or like short to high. Short wavelengths have a lot of energy. Yeah. If I expose you to gamma rays, I wouldn't do that. I would kill you. Okay, so you would die. They have super high energy. This is the kind of stuff that's emitted when like nuclear atom bombs go off. Okay.
You know all things.
Right?
Yeah.
So yeah, gamma rays. But even these things will take, look, X-ray, high energy, that's why when you go to the dentist, you know, they like put that lead thing on you. The reason why is because your reproductive organs are in here and you don't want to expose them.
Oh, that's why they do that?
Yeah.
Wait, what?
They're trying not to mutate your eggs. Thank you. And they do your teeth. Anyways, and even UV radiation, you can get skin cancer, you know, going in the sun. So all of this stuff is because of the high energy, they have the ability to mutate your DNA. Anyways, gamma rays is like really, you know, really high energy. But removing the visible light spectrum, this stuff is not dangerous, obviously. This is what we can actually see in. What's interesting is some insects can actually see beyond into this infrared area, but we can't. Anyway, so the 380 is the higher energy of the visible light spectrum, 750 the lower energy. Anyways, remember you learned, you know, your colors were do this. My kids sing some sort of song. They learned their like rainbow colors. I forget what it is. At the end of it, it's like, don't forget purple hue. Anyway, I was singing it. So we care about the visible light spectrum. So I'm going to move on here. Photosynthesis gets energy by absorbing wavelengths of light. And what they're really absorbing are the photons. You learned in physics that photons are weird because they behave like particles, but they also behave like waves, and it's just kind of odd. You remember learning that? Yeah.
But
Yeah.
plants that don't have green leaves, do they undergo like
like what?
like
They're, okay, yeah. They do have green hearts, though. They have green leaves. Let me show you a picture. Hold on. That's a good question. Just hold on. Wait, I don't want video. Okay, well, anyway. I just want, okay, I'm in the wrong thing. See, they have green leaves, look. It's just the red leaves are special to attract the pollinators, but they do have a lot of green leaves. So.
why don't they plant red again after like
Okay, so y'all are asking these really good questions, and we'll get to that in chapter 39. I'll answer that in like February, okay? Maybe even March. It has to do with something called long day versus short day plants. Poinsettias normally don't bloom this time of year, but because they can grow them in like a controlled setting and alter the amount of like UV light they're getting and also the amount of darkness they're getting, they can make them flower earlier. Anyway, it has to do with time of year, and they're pollinators.
My mom had this thing, I don't know what it's called, like a Midnight Express plant, and it only blooms like one day of the year at midnight. Why? So cool.
Because that's, I know, there's all, I wish we did our plant unit still, because there's all this cool stuff. Because whatever its pollinator is, we have no idea what it would be. Would only happen to occur at midnight one time a year. I don't know what it would be, but that's why. It all has to do, the reason flowers are produced is because of pollinators. Pollinators will come, eat something on it, take the pollen, and take it somewhere else. It has to all do with the pollinators. I don't know. All right, so chlorophyll, the pigment, will absorb, notice over here, over this graph over here is something called an absorption spectrum. What does that really mean? Do you remember how we use the spectrum is? Okay, and you put different pigments in there, and they showed different peaks at different colors of the visible light spectrum. The higher the peak is when they're absorbing the most, okay? So if you put chlorophyll A in there, you would get a peak right over here, and you get a peak like over here in the purpley blue area and also the red orange area, okay? That's where they're absorbing the most. They absorb a little bit down here in like the yellow orange, and a little bit more in this blue area. Chlorophyll B has a different peak over in the blue. But notice chlorophyll B absorbs all from the purple to the blue really. And then chlorophyll B absorbs more over here in the yellow orange. But notice both chlorophyll A and B combined, they absorb all wavelengths of light except for what? Green. You see that? So that's why if the chlorophyll is there in the plants, we see green, okay? Because it's absorbing everything else. Chlorophyll starts to deteriorate in deciduous plants. Deciduous plants are ones that drop their leaves during the fall. It starts to deteriorate relatively early on in like maybe sometime September or October, right around now. And when they go away, the carotenoids are the ones that are left. There's other pigments called accessory pigments, carotenoids and xanthophylls that are right up there.
Wait, what
Okay, that's a good question. The reason is because during the wintertime, the sun is not as strong and the days are a lot shorter. And so those plants basically go into a state of dormancy where they're still alive, but they're not photosynthesizing anymore. They're just kind of hanging out. They're using their stored energy, which they store a lot of their extra sugars in their roots, just to remain alive, but they're not really doing much. It's like they're sleeping, kind of like a bear hibernation. Yeah. To conserve energy during the cold months, where they wouldn't be getting a lot of, their threat of losing too much water would be higher than their threat of like being able to like get their energy and all that fun stuff. So that's why. But the carotenoids and the xanthophylls are there. Notice the carotenoids absorb in the purple blue, but the carotenoids don't absorb anything in the yellow, red, and orange. So that's why when the chlorophyll start to go away, you start seeing those other colors. It's pretty cool. The accessory pigments, their function is not to undergo photosynthesis or to help with that, but their function is to absorb excess light energy and dissipate it so the plant is not harmed. It's called photoprotection. I want to point out something in your book. While y'all are reading your chapters, there is I'm getting your textbooks next year. Sorry for y'all.
I like
I know.
about my textbook.
On page 191, sometimes you see these little green boxes that say inquiry, and you might be like, oh, I don't need to read that. Yes, you should. Okay? This is, they highlight different experiments that have happened, and this one in particular is something you need to know. Because a lot of times, like on the AP exam, they use data from this particular experiment, and they ask you to interpret it. Now I'm going to ask you to know this on the test. So don't ignore these boxes. Okay? I'll try to point out the ones that I know you need to know, but this is definitely one you need to know. Okay, I'll talk about it, not today, I don't have time, but I will talk about it tomorrow probably. But just don't necessarily ignore those green boxes when you don't think they're needed. Okay, let me say a couple more things and I'll stop. Actually, I'm just going to trade some pretty leaves. I don't want to go into this much. Yeah.
Oh, it's not tomorrow.
Yeah. The Veterans Day something, I'll show it to you.
Yeah.